other organizers of this phytochem series. It's really a pleasure to have the opportunity to share some of the work I've been doing with you guys. So, as Luke mentioned, I started my own group at the Hebrew University a year ago. So there's not really a lot of interesting things we can share yet. So instead, I'm going to talk about sort of my past research, both from my PhD work and also then the postdoc. And this is still relevant actually to what we're doing today with my group, because we're sort of continuing and expanding these two projects. So starting from the first story, so this is, as I said, my PhD work, which I did at the lab of Asaf Aharoni at the Weizmann Institute of Science in Israel. And this project was focused on gene discovery and metabolic engineering of betalains. So betalains are plant pigments. They're probably less well known than other plant pigments such as carotenoids and anthocyanins, but they're actually one of the three major classes of pigments. But as opposed to anthocyanins and carotenoids, which have been very well studied in terms of their chemistry and biochemistry, betalains have always been sort of lagging a little bit behind. And one of the reasons for this is probably because they're relatively rare in the plant kingdom. So carotenoids and anthocyanins, for example, are more or less ubiquitous, but betalains are only produced by one specific order of plants called the Caryophyllales, which includes around 11,000 species, including some very well-known ornamental plants and also a few cases of edible crops. And probably the most well-known example is the beetroot, the red beet. So this is a group of compounds which contains more than 100 different ones, which are usually categorized in these two groups: the red to purple beta-cyanins and the yellow to orange beta-xanthines. So what we were interested in mostly in studying these pigments was their biosynthetic pathway, which was only partially known at the time we started the project. So I can sort of run you through this simplified version of the pathway, where tyrosine is initially hydroxylated to give L-DOPA. L-DOPA is then converted to bethalamic acid by this enzyme called DOPA deoxygenase, or DOD in short. And then this bethalamic acid can spontaneously conjugate with amino acids to form the yellow beta-xanthines, or with cyclodopa to form this beta-cyanin, which can then undergo additional modifications and decorations like acylations and glycosylations. So at the time, the actual first committed step of the pathway was unknown. It was thought for many years that this reaction is catalyzed by a tyrosinase or polyphenol oxidase enzyme, but there was never really good evidence for this. So this was the first thing that we focused on. So we started out by generating, I would say, two different sets of RNA-seq data. One was a multi-species analysis of several betalain-producing species, where we sequenced both betalain-producing and non-producing tissues. And then we also focused on one of these plants called Mirabilis jalapa, or 4 o'clocks. And here we did quite an extensive RNA-seq data set, which included 24 different samples, mostly different flower parts, which accumulate betalains as they mature. And so we used these two data sets and gene co-expression analysis mostly to find the gene candidates for this first reaction. And this eventually led us to find an ortholog of what we found in Mirabilis jalapa in red beet. And this is a gene called CYP7686. And so by using both gene silencing assays like VIX, which you see here, as well as heterologous expression experiments, this led us to the conclusion that what is actually happening in the pathway is that CYP76 AD1, which was previously thought only to convert L-DOPA to cyclodopa, also catalyzes this first step of tyrosine hydroxylation. And then there's another redundant enzyme, CYP7686, which can only catalyze the conversion of tyrosine to L-DOPA, but not L-DOPA to cyclodopa. And so after sort of deciphering this last unknown step in the puzzle, this led us to think about the possibility of metabolic engineering of betalains in plants, and doing this in a stable way, because all the initiatives from metabolic engineering of betalain before always depended on feeding with L-DOPA because of the unknown first step. So as I mentioned, this is a really quite simple and elegant pathway where to get engineering of the yellow pigments, the beta-xanthines, you would essentially only need to express two enzymes, CYP7686 and the DOD. And to get the simple beta-thinins, betanin, you only need to express three genes, which would be AD1, DOD, and another glycosyl transferase. And so we constructed this vector, which we named PX11, which includes these three genes, the DOD, the CYP7681, and this glycosyl transferase. And we transformed this plasmid into several different plants, mostly solanaceous plants, just by chance, because that's what Asaf's group usually works on. And it was more or less successful in all of the plants that we tried, and we could see this accumulation of pigments, in some cases, even after a few days in the different calli. Another interesting thing we could see by doing LCMS analysis is that together with the betanin compound, which we're expecting to see, in some cases we could also see the presence of other metabolites. For example, here this acetylated betanin from Nicotiana glauca, and this, for example, is a compound which is not known to be naturally produced in betaline plants. And so we eventually managed to get mature plants from these different calli. So this, for example, was a microtome tomato. What you see here at the top left is actually unripe fruit. When it ripens, it gets this dark red color. And betalains are known to be strong antioxidants, so we use this tomato fruit to show this. And we could indeed see a 60% increase in antioxidant capacity of the fruit compared to the wild type plant. This was another example of I think a less well-known plant called Solanum linguum, a wild species, or European black nightshade. And this was actually really interesting in terms of the chemistry that we saw, because as I mentioned, the different species in which we engineered betalains, we could see additional compounds to the betalain. Usually it would be maybe 2 or 3 or 4 different additional compounds. But in this case, we could see around 40 different candidate betalains based on their absorbance. And this is from the ripe fruit. Out of this 40, we could more or less verify that these are betalains based on their fragmentation. And of these 28, about half of them have never been previously reported. And from those that have been reported, you could see, for example, a compound which you would expect to find in a prickly pear, which is malonylated betanin, or a compound which is found in pitaya, dragon fruit, like apiosylated betanin, and so on. So the plants that I showed you until now were all expressing this vector called PX11. And we were also interested to see whether we can engineer plants to produce, instead of mostly beta-thinins, to produce the yellow beta-xanthines, or to produce a mixture of both types of pigments. And you can do this by essentially using either AD1 or AD6, or the combination of both. So we constructed this PX12 and PX13 vectors and introduced them into tobacco. And as you can see here, you can get these different color phenotypes based, again, on the different gene combinations. And by LCMS analysis, we could verify that the different colors are because of the different ratios between beta-tannins and beta-xanthins. So other than giving really nice looking plants with beautiful colors, these transgenic plants are also a very, very good platform to study the putative roles of betalains in plants. And we only just started to touch on that aspect. For example, they have been thought to provide some resistance against certain pathogens. And we tested this with botrytis infection of the tobacco leaf. And we saw a very strong effect of around 90% reduction in the lesion size in the betaline producing plant versus the wild type. So betalains are actually used today as natural food colorants. They're sourced from red beet, and they're also known as pretty good antioxidants. And so there's a whole sort of a nutraceutical industry around these pigments. And so when we found these genes that catalyze the first reaction in the pathway, we saw the potential commercial value of this. And so we issued a patent application, and this application eventually led to establishment of this startup company called Phytolon, which are also situated in Israel. And what they do is they produce these compounds in baker's yeast for the use as natural food colorants. And betalains and the ability to engineer them in a quite a simple way has also led to the fact that they've been quite widely adopted as transformation markers and reporter genes for plant researchers. So this is just a couple of examples. So as I showed you before, we were able to produce them both in plants, in yeasts, and this is also BY2 cells and so on. And they were also later used, for example, as a marker for mycorrhiza infection. And also, of course, the most well-known example is this Ruby gene, which is now used by, I'd say, probably hundreds of different labs, which essentially expresses the same three genes, but under a polycistronic construct using these 2A autocleaving peptides. So moving forward to my second part of the talk, and this is the postdoc, the work I did as a postdoc at Professor Anne Osborne's group at the John Innes Center in the UK, in which, to say briefly, what we wanted to do here is to discover and characterize new defense-related chemistry in wheat. And what drove us to do this is that, quite surprisingly, although it's a very important crop in agriculture, there's surprisingly very little known about its specialized metabolism and specifically in relation to chemical defenses. And the importance of this is that wheat is also susceptible to various pests and diseases, which ultimately cause losses of around 1/5 of produce. When we started this project, that was back in 2018, the wheat genome had just been published. This is the common wheat or bread wheat genome. And so we saw this as a really excellent opportunity to explore specialized metabolism in these species. And one of the ways that you can do this is by looking at gene clusters. So I think the idea of gene clusters is probably familiar to most of the attendants of this talk, so I won't get too much into it. And if you look at the several dozens of clusters which have been identified and characterized to date, you will see that the vast majority of them encode compounds which have some kind of defense-related activity. This can be antibacterial, antifungal, allelopathic, and so on. So for us, this was a good indication that looking for defense-related gene clusters in wheat would be a viable approach. So this phenomenon of gene clustering has been really elemental in pathway discovery of many different compounds. These are just a few examples that I'm showing here. And interestingly, actually, some of the first examples of gene clusters that were found going back around 25 years ago were also of defense-related compounds in grasses. So, for example, dimboa, the benzoxazinoid that's produced in maize, adenosine, the antimicrobial produced in oats, and the momilactones, which are diterpenoids produced in rice. And in addition to being very helpful to facilitate pathway elucidation of known metabolites, gene clustering can also enable you, to some extent, to do a sort of untargeted discovery of new metabolites, where instead of looking or trying to elucidate the pathway of a certain metabolite of interest, you can go naively approach any assembled plant genome and look for interesting looking gene clusters with the hope that then they will lead you to interesting new metabolites and enzymes. And taking this approach is specifically useful in cases where the metabolites are only produced in very specific conditions or in very low quantities. So if you would take the more traditional chemistry-guided discovery, some of these metabolites would be easily overlooked. But of course, coming from the genomic side, this gives you access to these different metabolites. So this is a sort of approach that we utilize in our wheat work. And we started by looking at gene expression data. So alongside the publication of the wheat genome in 2018, this work came out from Cristobal Wawi's group at the John Innes Center, where they compiled a very large number of different RNA-seq experiments from wheat, including experiments that included infection with different fungal diseases or related biotic stress. And they composed this weighted gene co-expression network analysis. And so we use this network analysis as our starting material. And specifically, we analyze these different gene expression modules to find the ones which are the most highly induced by these different pathogens, both highly but also consistently induced by different pathogens. And then we focused on these top 5 modules to see if we can find any gene cluster candidates within these modules. And this indeed led us to find these 6 different clusters from 4 different types. I'll just say here that we were working with common wheat or bread wheat, which is an allohexaploid, which contains these 3 different subgenomes, A, B, and D. And so by looking at the composition of these different clusters, we could also predict what kind of compounds they might be producing. So for example, BGC1, which is found both in the A and the D subgenomes, we could predict produces a diterpene based on its gene composition. BGC2, the same. BGC3, also found in 2 copies, we could predict produces a triterpene. And lastly, BGC4, we predicted to produce a flavonoid. Now, importantly, these 3 different classes of compounds have already been implicated in plant defense and specifically in the grasses as well. And another thing worth noting here is that if you look at the gene expression of genes within each of these clusters, you can see that all or most of the genes are also co-expressed, which is a very important indication to see that these clusters are actually actively producing a compound. So the main interest and the main sort of core of the work that we did was the functional characterization of these clusters. So to understand whether they produce genes which are really active, enzymes that are working together to make different pathways and so on. And we use this really efficient pipeline, which is frequently used in Anne's group, which is based on agroinfiltration in Nicotiana bentamiana, specifically using combinatorial expression of different genes. then using mass spectrometry to analyze the results that we got from these experiments, and in some cases also producing these compounds in Bentamiana in a larger scale and purifying them so we can also get full structural assignment by NMR. So this pipeline eventually enabled us to elucidate the full pathways from these 4 different clusters. And what we found pretty much worked with our predictions, where BGC1 and 2 produce these different types of diterpenes. These are labdane-related diterpenes. Cluster 3 produces this triterpene compound, which we named L-arenosine. And cluster number 4 produces this flavonoid, specifically isoflavone, which is named tritacine. And for this talk today, I'll mostly focus on these 2 clusters. So starting with cluster 3, the triterpene cluster. So this was essentially quite a simple cluster, which included only this oxidosqualene cyclase or triterpene synthase, and then 3'450s and a hydroxysteroid dehydrogenase gene. And using this Nicotiana Bentamiana aggregate filtration system allowed us to reconstruct this pathway. And then by doing NMR analysis of the N compound, we could see that this is a isoboinol-derived triterpene. After working out the chemistry of this pathway, we're also interested to see if we can find any clue as to what it might be doing in the plant. Is it really a defense-related compound? So for this, we went back to look at gene expression, and we saw that the treatment of detached wheat leaves with different chemical elicitors like methyl jasmonate, salicylic acid, chitin, strongly upregulates the expression of the cluster genes. And additionally, we could detect L-arenosine in the wheat-induced leaves. And this was important for us for 2 main reasons. One is, first of all, to see that this is a real compound, because of course we reconstituted this pathway in an artificial system using this Bentamiana. So indeed, we can find that this is a compound that, first of all, is really found in wheat. And secondly, we could see that it overaccumulates after treatment with methyl jasmonate compared to the control, which corroborates its proposed role as a phytoalexin or pathogen-induced small molecule. Also, infection of wheat with 2 different cultivars, accessions of powdery mildew. One of them is a wheat-adapted variety, and another one is a barley pathogen, which is not adapted to wheat. We could see that in both cases, these isolates induce the cluster genes, but interestingly, the barley pathogen actually causes a significantly stronger induction than the adapted wheat pathogen. We could also observe this similar type of pattern by looking at data from a microarray expression from a paper that came out a couple of years earlier, where they infected wheat with 3 different pathogens with adapted and non-adapted accessions. And we could see here as well that in the case of the 3 different pathogens, the non-host interaction caused a more robust induction of the cluster. And so we could hypothesize that the adapted pathogens are probably able to repress the activation of this cluster, like they also are able to repress other defense-related mechanisms. So jumping now to cluster 4. So this cluster was slightly bigger and contained genes which were really classic for the canonical pathway of O-methylated flavonoids. So it included 2 chalcone synthases, a chalcone isomerase, 2 cytochrome P450s, and 4 O-methyltransferases. And using the same approach which we did for the triterpene cluster, we also cloned and expressed in different combinations in Nicotiana Bentamiana. And this again allowed us to reconstruct the pathway. And when we then purified and ran NMR analysis for what we predicted was the end product of this pathway, right here, we were surprised to see that this was actually a non-methylated isoflavone and not a non-methylated flavone like we were expecting. And so the pathway looks more or less like this, starting from naringenin, and then you have two O-methylations, then two reactions by the two P450s. The second one is, I would say, the interesting one. This is what you would call a isoflavone synthase activity, where the ring moves to a different position. And finally, another methylation here. And this is a sort of linear pathway, but there will also kind of side products based on promiscuous activity of some of the methyl transferases and also the P450. So why was it a surprise for us to find that this is an isoflavone? So isoflavones are a specific groups of flavonoids, which are mostly restricted to the legume family. And they're also quite well known for their health promoting properties. They have activity against different kinds of cancer, menopausal symptoms, osteoporosis, and so on. And this is mostly due to their structure, which is very similar to our endogenous estrogens. And so estrogen receptors are able to recognize isoflavone. And this is why they're also named phytoestrogens. And these health promoting properties have also made them a very sought after target for metabolic engineering. And so this new isoflavone synthase that we found in wheat is different from the isoflavone, which is known already from soy and from the other legumes, where in our case, there are two different reactions. So the first one is 2' hydroxylation by this first P450, and then you have the aryl migration, which is coupled with the C-ring desaturation catalyzed by this second P450, which is the isoflavone synthase. And in contrast, what you have in soy and the other legumes is, first of all, the aryl migration coupled with 2-hydroxylation, and then another C-ring desaturation, which is catalyzed by a gene which is not a P450, a different type of gene family. So we are hoping that this new isoflavone synthase that we found would be useful in metabolic engineering of isoflavonoids in general. But it's a bit early to know this for now. So again, in this case, we were wondering if we could get any clue to what this compound, this tritacine, is doing in wheat. So again, we looked at gene expression, and here as well, we could see that the cluster is very strongly induced by pathogen infection. In this case, this is the fungal pathogen Fusarium pulmorum, and also that the compound itself is highly accumulated after the infection. So maybe 30 or 40-fold more than the control. If you want to understand the physiological relevance of a certain compound, a phytoalexin, then of course you also need to see where and when it's produced in the plant. So we did this analysis here of different plant tissues. It's not I wouldn't say it's an exhaustive analysis, but we looked at around 7 different tissues. And from this, we could find that this compound, this isoflavone, is mostly accumulated in the leaf tissues, like blades and sheath. What also really interesting for us was to see that it's also accumulated in the dry grains, which of course, this is a part of the plant that we consume as humans. And so because of the health promoting properties of isoflavones, we wanted to see if there's any kind of nutritional relevance to finding these isoflavones in wheat grains. And I would say the short answer to this is no, because the amounts that we could find in the grains are very, very low. So if you look here, we could see that the compounds are mostly found in this outer layer called the bran. But even here, the amounts are about 0.1 microgram per gram. So just for comparison, soy example contains milligrams per gram. So around 10,000-fold more than wheat. When we looked at this with these wheat grains with mass spec imaging, we could also corroborate what we saw with the LCMS results that I showed you, where you can see that the mass for tritacine, 329, accumulates mostly in this outer bran layer. And this also coincides with this plausible protective role, as you can imagine, against different pathogens that could be on the grain. And this is similar to other bran-bound phenolics, which were found in other cereal grains. So if we want to show the defense-related activity of this compound in wheat, the smoking gun would be obviously showing in planta effect of resistance to disease, right? So in this case, we were working with knockout mutants from an existing tidal population. And we worked with mutants for 3 different genes from the pathway. And the idea here was that if this is indeed a defense-related compound and you obstruct its production, then the mutants should be more susceptible to disease. And so we tested this with 3 different pathogens, Fusarium palmarum, powdery mildew, and cereal blast. But in none of these 3 pathogens, and for none of the mutants that we looked at, we could not see any observable difference in susceptibility between the wild type and the mutants. But of course, disease resistance is a complex quantitative trait which is affected by many genetic factors. And so we also wanted to see if we can assess potential antimicrobial activity of these compounds in vitro. And here we saw very clear results, both in antifungal activity, specifically, you can see here, inhibition of spore germination and tubule ingration in botrytis, but also inhibition of bacterial growth in liquid media. And we saw this with several different bacterial strains that we tested. And in some cases, we could see quite potent activity. For example, this Staphylococcus aureus was inhibited in concentrations as low as 1 micromolar of tritacine. So going back to the cluster itself, as I mentioned, this cluster is found on the beet subgenome of wheat and not on the AM subgenome. And correspondingly with this, we could see that this cluster is also conserved in the wild grass Aegilops tauschii, which is the donor of the D genome, but it's not found in the genomes of emmer wheat, which is the donor of the A and B subgenomes. And doing metabolic analysis of Aegilops tauschii and also several different varieties of emmer wheat, we could also see that this compound is also produced in Aegilops tauschii and missing from emmer wheat. And so what we can say about this is that hexaploid bread wheat, which was formed around 9,000 years ago, it was formed through this natural hybridization event between the domesticated emmer wheat and Aegilops tauschii. And so what we can say is that this tritacine cluster was introduced into hexaploid wheat through this natural event. And I think this is a nice example of how hybridization-derived polyploidy can lead to a metabolic enrichment of a certain plant species. So to summarize this part, we used an untargeted approach, which allowed us to rapidly discover and characterize these new defense-related metabolic pathways in wheat. And now what's really interesting for us to explore is to really understand if and how these different compounds that we found contribute to wheat resistance against different pathogens, to which pathogens and which parts of the plant, and so on. And hopefully this information can be used in the future to develop more resistant varieties. So before going to acknowledge all the people that took part in these two projects, I also wanted to acknowledge this really incredible plant species, Nicotiana bentamiana, which is again probably known to most of the attendants of this seminar series. And what was really great for us to see is that we were able to produce these compounds in bentamiana, but also to produce them very efficiently. So the amount of product that we got allowed us to easily recognize the peaks we were looking for. And this really only demanded very little intervention with Nicodena betamiana. So we used like different strategies for example, for producing the triterpenes, we co-express with our gene this feedback insensitive HMG reductase, which has a truncation. For flavonoids, we also co-expressed a transcription factor from aldehydoxys, which upregulates the phenylpropanoid pathway and so increases the precursor availability. And for the diterpenes, we expressed what would be usually plastidiol enzymes by truncating the N-terminus. They were stuck in the cytosol. So to acknowledge, again, all the different people that took part in this first story that I told you from Asaf Aaroni's group at the Weizmann Institute of Science, and also other collaborators at the Weizmann Institute and from the Hebrew University of Jerusalem. And for the work that we did with wheat at Ann Osborne's group. And here I'd like to acknowledge mainly Rajesh Chandra Misra and Amel Demerdash, which are two other postdocs from Ann's group, which closely collaborated with me on this project, as well as our collaborators from the John Innes Center and the Norwich Research Park and the funders of this project. And also I'd like to acknowledge all the guys in my recently established group at the Hebrew University of Jerusalem. So as I mentioned in the beginning, we're still actually active in these two stories, the betalains and wheat phytoalexins, as well as phytoalexins in other cereals. And so if anyone has similar research interests, please welcome to get in touch. And I'll stop with this. Thank you.
Thank you very much. Great presentation. For questions, you can just raise your hand. You can post them in the chat. You can just unmute yourself and ask your questions. Please do so. Yeah, I might start with a question. I have a more general question to your, I guess it's mostly the first part, but also for the other parts. I'm interested in when you characterize these metabolites, it seems like that often you are actually performing NMR analysis later. So are you like using this large scale bentamiana expression to produce enough of this compound and then having like some sort of automated pipeline for purifying these compounds?
Yeah, sorry, I probably should have mentioned and explained that better. Yes, what we would do is essentially repeat the same experiment that we would do with manual infiltration by syringe into bentamiana in a larger scale, and we would use vacuum infiltration to do this. And then we would infiltrate something to the extent of 100 different plants, which would give us, in most cases, somewhere between several milligrams to tens of milligrams of compound, which would be enough to do NMR analysis. And in our case, we got sort of two birds with one stone because we could also use these compounds that we purified to do in vitro activity assays because we wanted to find out more about the activity of these different compounds.
All right, yeah, 100 plants. That's quite a few. Thank you. Yeah, we have two questions in the chat. The first one is from Liz. Can you comment on the prevalence of the prevalent, how prevalent you think biosynthetic gene clusters are in plants?
That's a really good question. And as far as I know, there isn't a clear answer to this. Definitely, it's not the majority of biosynthetic pathways are not clustered, but on the other hand, it's also not that rare or exceptional. So there have been already a few dozens of gene clusters which were characterized, probably I would say maybe 50 or 60 or so. And if you use these sort of algorithms to look at or to look for gene clusters in plant genomes, so usually you would find around again tens of gene clusters in each genome that you look for. In wheat specifically, because it's a hexaploid and you find a lot more, I think it was somewhere around 170. But here it's very important to note that most of these clusters are only cluster candidates and they would be inactive for the most part. So either not expressed or the genes are pseudogenes, mutated, and so on.
Thank you. We have another question in the chat. What do you think is the primary function of beta lanes?
The primary function is, I think, the thing which is probably the most obvious to us is as to attract pollinators, right? So if we see it in the flowers, it gives color. And actually, interestingly, the beta-xanthines are also fluorescent, which is also important for pollinator attraction. Also, colors in the fruits can attract herbivores. So this is a sort of obvious role that betalains have. But as I mentioned, they're also known to have these different functions. So to some extent, they can provide protection against abiotic stress. So for example, excess UV light, but also against biotic stress. So this is probably due to their antioxidant capacity.
Okay. Yep. Yes, please, Jörg.
Great, thanks, Lars. That was a fantastic talkie. Really, really great stuff. And beautifully illustrated as well. Can't believe the volume of work you presented there. Regarding the various plants that you engineered to produce and express betalains, including tomato, this is a two-part question. So the first part question is, have any of those been commercialized for production consumption?
The short answer is no. So when we were sort of involved with this patent applications and what we can do with it, so of course we were very interested in this aspect as well as so one potential application, for example, could be generating new varieties of ornamental plants, right? So different flower colors and so on. But I think we were eventually convinced that this would not be possible or would be very difficult because of the regulatory aspects of GMO, right?
Okay.
So instead, eventually the route we went down was this production of these pigments in yeast.
Okay.
It's natural colors.
Then it becomes a quick three-part question, but last there will be short questions now. So Katie Martin and her teams, they released, I believe, a flavonoid producing tomato, at least in the US. So how did they get around the regulatory concerns and you would not?
That's true, but I think it actually took them a very long time. So
okay. Yeah.
The paper came out maybe 2008, if I'm not mistaken. So it took a long time. And again, when we were, this is back in 2016 and 17, I think the purple tomatoes from Kathy's group wasn't yet approved. So we didn't have this tree to hang on to.
Okay, good. So, yeah, my third question is a semi-serious one related to engineering crops and plants and all sort of things for better lean production, coming from enjoying time in my kitchen. When I make tomato sauce and I mess around with carotenoids, they're very easy to clean up and get out of the laundry and so on. But when I do anything with red beets, it's a huge mess, and sometimes I can't get it out of the cutting boards and out of the cloths and so on, and the dishcloths. What's the issue there? Why do they slice
Wow,
heavily?
I wish I had a really good tip of how to get rid of phthalates.
Yeah.
Um,
I think it
I
makes
don't.
yeah, I wonder how this is being dealt with when you commercialize heavily betterling producing food ingredients, and
Mhm.
how consumers deal with this. Because
I
again,
think the best thing is probably
yeah,
not to eat them when you are wearing a white t-shirt.
yeah, I would not recommend it.
Yeah,
Okay,
otherwise I can't think of a specific hack to
yeah.
get rid of it.
And again, this was not the most serious question. The first ones were more serious about the science. Fantastic work, really beautiful. Thank you.
Thank you very much.
Yeah, we have one more question in the chat from Pavan. In your fungal assays with betalains, you have checked only PYK11. What about the other PYK12 and PYK13, for example?
If we check them with relation to resistance,
Yeah, for resistance, have you checked that because you are producing both beta xanthine and beta alanine? So is the ratio of that these two compounds are affecting the fungal in a pathogen in a provide
uh,
tolerance to pathogen?
yeah, that's an interesting question. First of all, we didn't try this, and there's actually very little known about this antifungal, antimicrobial activity of betalains in planta. I can't think of any other example except for what we showed with this PX11 tobacco. So really, the knowledge about the mechanism and which of these compounds, the beta-xanthines or beta-cyanins, have more or less activity, I think it's not known, but definitely an interesting thing to explore. And as I said, these transgenic plants that we generated, I think, are a really good system to explore in general the roles of betalains in plants, because you have this clean system where you can compare producing versus non-producing plants. So this is one of the things we're still interested in.
Thank you.
Yeah, Hiroshi.
Hi, that's great talk, and
Hi,
congrats
Hiroshi.
on your new position.
Thank you.
Yeah, I'm just wondering if when you express the first construct with CIPS 7081, with still downstream, did you detect the beta xanthine?
Yes, there are beta-xanthines there.
Oh, okay.
There are beta-xanthines. There are, I think the quantity is much less than the beta-cyanins, but definitely they are there.
Oh, okay. Okay.
And yeah, so this is due to the fact that the bethalamic acid has spontaneous conjugation with whatever amines are in the area.
Okay.
So yeah, we did see them.
I see. Yeah, when we express ruby, just we didn't see it, so I wonder if you saw the same thing. But
In which plant?
let me see. Nicotiana.
In Nicotiana metamiana?
Yeah.
I can't remember exactly there. We did produce a transgenic bentamiana. I can't remember what we saw there, but I think it will be in the paper probably.
Okay. Yeah, I thought there may be some additional channeling happening in the pathway, but yeah.
Yeah, that could be.
Right.
The bentamiana was actually very interesting. It was the only plant that gave us a very sort of detrimental phenotype. Don't know if this is what you see with the ruby, but with the other solanaceous plants, they produce betalains in very high quantities and they looked mostly happy, but the bentamiana was completely sort of mutilated and almost male sterile.
In the stable lines, right?
In the stable lines, yeah.
Yeah.
You were talking about the agroinfiltration?
Right, right.
I can't remember.
Okay.
Need to look that up. Sorry.
No worry. Thank you.
Yeah, do we have more questions? If that is not the case, I would like to thank Guy again for this wonderful presentation and the nice discussion. And I hope to see you all again in 2 weeks. our next Phytochem talk session.
Thank you very much.
Thank you.
Thank you for the invitation again.